Hello, welcome to this introduction to ChEMBL. My name is Prudence Mutowo. I'm a bioactivity data manager with the ChEMBL database. The URL for finding ChEMBL is as you can see on the screen. And if you wanted to contact the database staff regarding any queries, help, or any data errors, please contact us at chembl-help@ebi.ac.uk. The topics I'm going to cover today include what ChEMBL is, what it's useful for, and also where the information in ChEMBL comes from, how we process that information, and how, as a user, you can search ChEMBL using the web interface to get relevant information that you need. If we start by talking about the utility of ChEMBL, we provide insight, tools, and resources for drug discovery. And this slide shows you some of the examples in which ChEMBL data has been used in the field of drug discovery. The first is within the Open Targets platform, where small molecule information in ChEMBL is used to make disease associations between diseases and their relevant targets. And if we come to the center, it's an example of a publication giving an overview of the landscape to the drug targets for approved drugs, for FDA approved drugs, encompassing a variety of protein targets and protein target classes. We also have researchers who sequence genomes and come to ChEMBL to potentially identify small molecules that can be used to target the newly sequenced genomes in a view to fighting neglected tropical diseases. And if we move on to the actual database content itself, ChEMBL is a manually curated database, and the information comes primarily from scientific literature. We extract information from main medicinal chemistry journals, such as the Journal of Medicinal Chemistry, Bioorganic Medicinal Chemistry Letters, as well as Science. We also have information from journals that carry absorption, distribution, metabolism, and excretion relevant information, as well as agrochemicals. With an interest in toxicity, we have information from reviews, such as those covering hepatotoxicity and cardiotoxicity. We're in a fortunate position to have some data sets given to us by organizations freely and that you can access. And some of the organizations that contribute to our deposited data sets include the Medicines for Malaria Venture, MMV, Drugs for Neglected Tropical Diseases, DNDI, as well as pre-competitive data from pharma, such as AstraZeneca and GSK. From public databases, we take information from PubChem, and the information we take from them is confirmatory assays. We also take information from the Binding DB database, which is a manually curated database, and the information is curated from patents. So we take the bioactivity information from the patents. Other reference data sets that we have include toxicity relevant sets, such as those from OpenTG Gates, the Tox21 projects, as well as Drug Matrix. On the compound side of things, we have clinical research compound sets and approved drugs from the FDA, as well as information from clinicaltrials.gov, the World Health Organization, and USAN. On the drugs and small molecules, where possible, we try to provide information such as black box warnings and withdrawals, therapeutic mechanisms and indications, as well as annotating molecule features. And so if we come to ChEMBL and have an overview of the current state of play, this is from ChEMBL 22, we have about 92% small molecules and 7% peptides. Currently, the information is extracted from 65,000 papers across 47 journals. In terms of compound records, we have just over 2 million compound records, of which 1.6 million of these are distinct compound structures. Our over 4 million activities come from assays such as binding measurements, functional assays, and ADMET. And on the target side of things, we have just over 11,000 targets, of which 5,400 of these are protein targets, and we cover over 2,500 human targets. Coming to our actual workflow and structure, like I said in the beginning, the majority of the information comes from journals. So we have curators who will read and extract information from a publication. Some of the information that's extracted is the kind of assays, and from capturing the assay information, we go on to perform a target assignment step, where from the publication, we will then work out what the target in question for the experiment was. So say, for instance, if it's a protein, such as phosphodiesterase 5, PDE5, we would capture the information. In cases where it's cell or an organism, we would take that information as well, and then perform a next step, which is a target registration step, which allows us to assign comprehensive and concise alphanumeric database identifiers. So in the case of PDE5 protein, for example, we would use the UniProt accession of that protein, capture it, and include it in the information relevant to the assay, so that users can come to us and query using concise identifiers such as UniProt identifiers. In the case of cells, for instance, we would use a cell dictionary. Similarly for tissues. And for whole organisms, we would use the NCBI taxonomy to capture the organism information. And we also use a variety of other ontologies to make the information more concise and easy to retrieve. One of the main ontologies we also use for targets is the gene ontology, and we use Go terms curated by the Gene Ontology Consortium to capture the processes, functions, and cellular distributions of our protein targets. And all this information is deposited in the ChEMBL database under a series of tables, including tables such as the target dictionary, which would have the target information. If we move on to compounds, a similar kind of process where the compound structures are identified and drawn by our curators. This is followed by a standardization step, including processes such as salt stripping or salt removal, as well as the creation of mol files and establishing of a hierarchy where we have parent structures and their related child terms. And onto the parent structures, pertinent properties are calculated, which are useful and informative in the areas of drug discovery, such as the A-log-Ps, hydrogen bond donor and acceptors, the molecular weights, as well as parameters such as the QED, the quantitative estimate of drug likeness. A similar kind of process occurs where after a compound registration and assigning of a unique ChEMBL ID for each compound, this information would then get deposited in the ChEMBL database under a series of compound-centric tables. If we come on to the activities, in cases where activity information exists in the paper, we would extract this information and capture the units represented as well as the activity measurements. We also have a second step in which we carry out an activity curation process where we standardize the units. So say, for instance, if the same activities are recorded using different units such as micromolar and nanomolar, we would choose a standard unit and then represent similar activities using the same kind of unit to make it easy for people to come and query activities reported in different papers, for example. We also have a system where we flag outliers. So if certain values reported in the publication are higher or lower than the expected, we would still capture the information and have the publication as a reference, but we would flag it to say it is out of the range expected. And again, this information is deposited in a series of tables that you would find in the ChEMBL database. If I can say a little bit more about the ChEMBL targets, we have two main groupings of targets, mainly molecular targets and non-molecular targets. And this target grouping represents the kind of targets that we find in the publications, in the papers that we manually extract and curate. On the non-molecular targets, if I start at the bottom of the slide, we have targets such as cell lines, subcellular fractions such as mitochondria. For example, it might be a tissue target such as a liver or a whole organism such as the rat. On the molecular side of things, we have protein targets, we have nucleic acid targets. We also have protein nucleic acid complexes as a target class, and in some cases a small molecule is also a target in the assay context. I'd like to say a little bit more about the protein targets. Because this is by far the largest target class that we have, we try and break it down into more informative categories, such as whether the assay was performed to a single protein, example being PDE5, or whether it was a protein family, such as the family of muscarinic receptors, or in cases where the target was a protein complex or a protein-protein interaction, as you can see highlighted there by the P53 MDM2 example. And this is where you can find us. So if you were to type that URL into your browsers, this is where you would find ChEMBL. And we have two main functionalities, the top search functionality, as well as the sidebars, which are a series of tabs that can lead you through to more precise or more specific information, depending on what you're looking at. So if you start with the tab to your left-hand side, is a download tab where you can download the information, the database itself if you'd like to. We link out to a resource called UniChem, which allows you to map chemical identifiers across a variety of chemical databases. There's also a tab to our ChEMBL patent database and the malaria and ChEMBL neglected tropical diseases datasets. We also have information on the statistics for each of the ChEMBL releases. So our latest version there, ChEMBL 22, will give you information on the number of targets, the compound records, and so forth, as well as a full release notes description. And at the very bottom of that, you have the ChEMBL blog, which advertises things like ChEMBL webinars and any other news that you might like to keep up with about the goings-on at the ChEMBL database. If we look at the top tab, you can search ChEMBL using 5 main categories, namely compounds, targets, assays, documents, cells, and tissues. If you were going to search the interface that I've just previously shown, you can search either using a compound name or a structure. So you can have a trade name, a brand name, or a structural name. On the target side, you can search using a protein target name, you can search using a FASTA sequence, an accession, or an ID. And on the assay side of things, you can search using an assay description such as cardiotoxicity. For literature, you could search using the author name, or the title, or keywords from the abstract. And so if we start searching ChEMBL for a particular compound and we wanted to get the compound information from the main website, we could search from that main text bar and type our compound name in, or we could take the structural information that we have and draw our structure and then search using a substructure or the full structure, or we could also search using a list of identifiers or keywords that relate to our compound. We could also use smiles as well. So in this right-hand column, you can have a list search where you can search using smiles, or the ChEMBL ID if you know it, or the keyword, which might be the trade name. And if we were to search ChEMBL using a keyword such as caffeine, and by clicking on compounds, we would get all the relevant compound information relating to the keyword search caffeine. We would get a series of compound structures, as well as their synonyms where the information caffeine is found. And if you wanted to download all this information and filter it, if you come onto the right-hand side, under this Please Select tab, you could download all the information. A similar search on bioactivities or clicking onto the bioactivities would give you this Excel spreadsheet type view, where we could then have information for the compound that we've searched for or the keyword that you searched for, and the activities information available in the database, where we give you the activity types, whether it's a log P, whether it's a stability, as well as the values and the assay type that it is, as well as a description of the assay itself, give you information on the source, where the information's come from, There's also a reference set which could link you directly to the journal or to the data set that the particular assay is associated with, as well as the assay target, where the assay comes from. So is it from where the assay organism is, whether it's human or it's rat, for instance, and the target type. Is it a protein target? Is it a whole organism? And you could download the bioactivity information as an Excel spreadsheet to do further filtering or to plug into a next tool to do further analysis. You could also download the structural information as an SD file. If we move down further onto the compound report card, which is the view that you would see if you were to type in the ChEMBL identifier or a name and then click on one particular compound. You would then get information on the ChEMBL ID itself, the compound ID, the name of the compound, synonyms that we've captured to the compound in the database, as well as a structure. On the compound structural representations, there's a tab there that gives you how you could download the mol file, the SMILES, the inches and inchy keys, as well as the different forms of the compound in ChEMBL, as you can see over there. And from these set of icons, we have descriptors of molecule features. So say, for instance, if you were to hover over this in the interface, this is the little icon that tells you that this is a synthetic small molecule versus, say, for instance, a natural product. And these are informative that they tell you information like the route of administration, the mode of administration, whether it's prescription only, whether it violates parameters such as the Lipinski's rule of 5, and if there are any associated black box warnings or warnings of some kind of toxic effects on administering. We also have a panel that gives you the mechanism of action of the compound. And in this case, the compound we're looking at, lapatinib, has been shown in DailyMed to act as an epidermal growth factor receptor ERB1 inhibitor. And this is the information on the targets. New in ChEMBL is a little widget that allows you to look at the metabolic pathways for the particular compound that you're looking at. So for the compound we're looking at here, lapatinib, we can look at the different ways in which the compound is metabolized. And by highlighting over this, you'll have information on the enzymes that are involved in the metabolism of this compound and the various metabolism byproducts. Moving further down onto the compound report card, we have two main summaries for you. The first summary is a bioactivity summary, which will give the activity types for the compound we are looking at under the different classes shown on the legend. And both the legend and the pie chart are interactive. So if we wanted to click on and get a subset of the activities, say for instance the ones that are IC50, we could click either on the legend or on the subsection on the pie chart to get a snapshot of the IC50s. Or we could download using this button the full activity type for the compound in question. Similarly for assay summaries, grouping of all the assays for the compound in question. We have a section here on target predictions, and this is a piece of work that was done to try to identify other targets that might be useful for investigation against the compound that we're looking at. And in this slide here, I have a detail of a blog post that was done giving you the information as to how these predictions were generated if anybody wanted to go away and have a further play. We also have information on things like compound cross-references, so other compound-centric databases that we would link out to that have information for the compound you're looking at, such as ChemSpider, DailyMed, and PubChem, as well as the World Health Organization Anatomical Therapeutic Classification ATC system. On the other hand, if you wanted to come into ChEMBL and you had a target that you were working on and you wanted to get target-specific information, similarly to the compounds, you could search using a keyword, such as in the top bar, mescarinic, or you could use an accession or gene symbol on the keyword search tab in the middle of the page, or you could come in with a FASTA sequence, enter your sequence in, and then perform a blast search. Similar to the compound report card, we have a target report card that has a unique ChEMBL identifier for each target. We tell you what kind of target it is. In this case, it's a single protein, its preferred name, synonyms, the organism from which it is obtained, as well as a protein target classification. So in this case, we're showing the protein family. So what family does my target belong to? Other target components and target relations. So are there any other entries in ChEMBL where this target is found? And in the case of the one that we're looking at, the M1 receptor, yes. It's also found, for instance, as a selectivity group with M1 and M3 receptor components, or as a chimeric protein where you have the M1 and M5 chimeric protein target. Similar to compounds, we also have pie charts that capture summaries of the activity and assay information that we have. So in this case, for activities, for the M1 receptor, you can click on either the pie chart or the legend to get subsections of information, or you could download everything for the target of interest. We also have a useful little plot for you here, which is a target ligand deficiency plot, which essentially, for the target that you're looking at, tries to color code or give information about the ligands the target has been reported for, and also how efficient or how good these ligands are at binding the target. And this plot has been generated using two parameters, the surface efficiency index, SEI, and the binding efficiency index, the BEI. And the coloring system there, you can get full information on that by clicking on this little info button highlighted in red on the interface. But suffice to say, we're trying to give you a quick overview of those ligands which might make good potential small molecules for the target that you're looking at by using a traffic light system coloring, where the green, the dark green, are proposed to be ligand deficient, whereas the red are not so. And so you could download this information again if you wanted to do a further filtering, or you could click on the button to get an indication of exactly how we came across the coloring and the calculations. As you move down the target report card, we also group the compound information relevant to the target using drug discovery parameters, important parameters such as molecular weight. So we would have this bar chart here that shows you the distribution of the molecular weight for compounds for this target. So if you wanted to take a snapshot and only view those molecules, for instance, that were less than 500, we could find on the bar here, you could click on those, or you could download everything. Similarly, for parameters such as A log B and the polar surface area, a similar kind of designation. On the target cross-references, we cross-reference out to databases that have relevant target information, such as Array Express and Ensemble, as well as the gene ontology information from Go, which would give you an indication of the kind of processes or functions or locations the target you're looking at is involved in or is a part of. We also reference out to other databases with target-centric information such as CanSAR and the Human Protein Atlas and UniProt. And on the target structure or sequence, we can have information from Interpro and PFAM, where you can get a better handle of the domains or motifs that your protein target might have by going to the corresponding Interpro entries associated with your target. If you were to perform a blast search, I mentioned earlier that you could come in with a sequence and perform a blast search to your sequence. You would have a result that would show you percentage identity of your target versus other targets deposited in the ChEMBL database. So you'd have information on the identity there, but you'd also have information on the names and the target types and the organisms for sequences similar to your target of interest, as well as the number of compounds for the targets that show similarity. And so you could sort these by ID or significant, for example, or organism if you wanted to, and then filter or drill down further to identify other targets of interest. We have a series of hierarchies in ChEMBL that allow you to navigate better the targets that we have. And we have three main hierarchies. We have a protein target tree, we have a taxonomy tree, and a gene ontology tree. The protein target tree is a classification of the proteins in ChEMBL in the four main families of drug discovery, as well as any other families where we have natural classifications for. And you can navigate this tree either by coming onto the hierarchies already established for you, clicking on them, and finding out which of the proteins are involved in a certain family, for instance, such as epigenetic regulators, and then lead out to the compound information. Or you could simply perform a keyword search here and type a particular keyword such as membrane, and then get led to the families that we have that have membrane as the keyword. Similarly, on the taxonomy tree, you could either come to the tree itself and start to browse the hierarchies and be led to targets that are found, for instance, in the nematoda class, or we could type in an organism name and be led to organisms that are found in the target class. For the gene ontology, a very similar system occurs here, where you could either navigate the tree on the three aspects of GO, molecular function, biological process, or cellular component. Or indeed, you could type in a keyword, such as here, immune, to find out all the GO classes that contain the keyword immune, and then drill down to proteins that are found in immune system process by virtue of having GO annotations that belong to this class. And other ways of accessing ChEMBL besides the web interface are we have web-based GUIs, we have web services, we have an EBI RDF platform, as well as those downloads there, which you can get from the ChEMBL site and navigating onto the FTP site. And we have tools such as MyChEMBL that allow you to download the database onto your own space, and third-party platforms and databases such as OpenFacts, BindingDB, and PubChem. We link out to a variety of internal and external resources, internal to the organization that we're housed in, the EBI, and externally to other groups and organizations working in the field of drug discovery. And that's just an indication of some of the resources that you would find cross-referenced within our web pages. And at this point, I'd just like to say thank you very much. And if you do have any questions about any of the material that I have presented, please can you contact us at chembl-help.ebi.ac.uk, and we'll try as much as possible to help you.